Dear students, this video is regarding the synthesis, storage and release of noradrenaline, and myself Dr. Deepa from the Department of Pharmacology under the designation of Associate Professor. The specific learning objectives in this video is about the synthesis of noradrenaline, the storage of noradrenaline, how it has been released in our body, how the action of it gets terminated, and what are the fate of these neurotransmitters. How the noradrenaline is being synthesized in our body. So, noradrenaline has been synthesized from the phenylalanine, which is an essential amino acid in our body. So, this phenylalanine get converted into the tyrosine in the liver under the influence of tyrosine hydroxylase enzyme. Then this tyrosine, it gets transported into the adrenergic neurons under the influence of tyrosine hydroxylase enzyme, okay, and it gets converted into dihydroxyphenylalanine, and this is the rate limiting step in the synthesis of noradrenaline. And this dihydroxyphenylalanine is then converted into dopamine in the cytoplasm under the influence of dopamine dihydroxylase decarboxylase enzyme, and this dopamine, it is then stored inside the vesicles, and in the vesicles then dopamine which has been stored get converted into noradrenaline under the influence of the beta hydroxylase enzyme. And once it has been formed in the vesicles, it has been undergo methylation under the influence of N-methyltransferase enzyme. So this noradrenaline get converted into adrenaline in the adrenal medulla cells. So this is how it has been synthesized in our body, phenylalanine to adrenaline. So once it has been synthesized, it will be stored in the vesicles, and we'll see how it has been released from the vesicles. So once it has been stored into the vesicles, inside the vesicles it will be stored as dopamine, and then dopamine gets converted into noradrenaline. That's what we have seen in the previous slide. And then this noradrenaline, under the influence of N-methyltransferase, it forms adrenaline in the medulla. So how it has been released from these vesicles? Whenever there is any action potential which has been generated in the nerve endings, that will open the voltage sensitive calcium channels. When the voltage sensitive calcium channel is being opened, there will be increase in the intracellular calcium. This increased intracellular calcium results in fusion of the vesicles with the surface membrane, and there will be expulsion of noradrenaline with the co-transmitters. So along with the co-transmitters and dopamine beta hydroxylase enzyme, and this can be blocked by bretylium and guanethidine, and this can be enhanced, that expulsion of noradrenaline can be enhanced by tyramine and ephedrine, okay, and this released noradrenaline then acts on the adrenergic receptors which are present pre or post synaptically. So this is how it has been released from the vesicles, and what happens once it has been released. So next is about the various inhibitors which acts on the specific steps in the synthesis, and also how they inhibit the action. So first is about I told about the rate limiting step. The rate limiting step is the conversion of tyrosine to dopa. So tyrosine under the catalyst of tyrosine hydroxylase enzyme. So this is being inhibited by met-tyrosine. So this is the rate limiting step where the met-tyrosine inhibit that conversion of tyrosine to dopa. Then I told about the decarboxylase enzyme. Okay, so the conversion of dopa to dopamine under the influence of dopa decarboxylase, that is inhibited by carbidopa. Okay, and then the conversion of the dopamine to norepinephrine, that is by inhibiting the dopamine beta hydroxylase enzyme, that is by the reserpine. So another inhibitor, that is in case of the norepinephrine uptake, that is uptake 1 into the cytoplasm. So norepinephrine uptake into the cytoplasm is inhibited by cocaine and tricyclic antidepressants, and even norepinephrine serotonin reuptake inhibitors also inhibit this. So these inhibit the uptake 1, that is the uptake of norepinephrine into the cytoplasm. These are the various inhibitors that inhibit the synthesis of this norepinephrine in the body. Next is about the termination of action. So once it has been synthesized, once it is released from the vesicles, okay, I told they act on the receptors. So they also know how the action gets terminated. So by the diffusion out of the synaptic cleft, it undergoes metabolism. Okay, and transportation into the cytoplasm. I told about the reuptake 1, which has been inhibited by cocaine and tricyclic antidepressants. So that is one way of getting metabolized. Another is transportation in the post-junctional cell. That is reuptake 2. That also a kind of metabolism what is happening. And final metabolite that is of norepinephrine and epinephrine is metanephrine and vanillyl mandelic acid. So these are the metabolic products or the end products of this norepinephrine and epinephrine. So that is under the influence of the enzyme monoamine oxidase. Monoamine oxidase enzyme is the enzyme which is responsible for the formation of this metabolites, and catechol O-methyl transferase. Catechol O-methyl transferase. So once you measure the level of the metanephrine and vanillyl mandelic acid, so that's how you know how much this synthesized, it is being synthesized, and how much it is that getting turned over in the body. So once it has been released, it acts on the various adrenergic receptors that will be present in our body. So about the different types of adrenergic receptors and what are the actions mediated through these adrenergic receptors, that will be the next video. Okay, so that's about this video, the synthesis, release and the metabolism of this catecholamines. Okay. So thank you. Have a wonderful learning.